my name is Emily Pavlovic, and I am a current graduate student at the University of Minnesota Duluth in the Integrated Biosciences program. Today, I'm going to tell you about the research that I'm doing for my master's thesis, which is centered around improving our knowledge of migratory connectivity for raptors at Hawk Ridge. So many of you, if you're from Minnesota, have probably spent some time at Hawk Ridge enjoying the incredible sight of migrating raptors in the fall. But for those of you who haven't, or maybe aren't familiar with Hawk Ridge, it's located in Duluth, Minnesota, along the basalt ridges, and it is a hotspot for raptor migration, mostly due to its geographic location in relation to Lake Superior, which acts as a sort of funnel for migrating birds. Counting and banding of raptors during fall migration has occurred pretty consistently since 1972, and on average, each fall, about 76,000 raptors are counted. So that's an amazing raptor migration. And to give you just kind of an idea of what that means for different species, I've put the fall 2020 counts for some species up here in this table. So you'll see about 33,000 broadwing hawks were counted this fall, almost 14,000 sharp-shinned hawks. So just an absolutely incredible migration. In addition to the count, thousands of raptors are also banded each fall. And so this combination of counting and banding has led to an extensive long-term data set that we can utilize to ask very interesting questions about raptor ecology and migration. However, one very important part of the story is missing, and that is, where are these raptors coming from? We know that most are coming from northern Minnesota and Canada, but where exactly and how far they're migrating is not really well understood. So that's the goal of my research, to create that link between the breeding grounds of raptors and their passage through Duluth, and in doing so, to improve our knowledge of migratory connectivity. So what is migratory connectivity? Well, migratory connectivity is the linking of individuals or populations between different stages of the annual cycle. So this includes between breeding locations, any places that are utilized during migration, as well as the areas that are used during the non-breeding stages. And this includes both spatial and temporal information, so both information about the timing and the specific geographic locations that are utilized. So why is migratory connectivity important? Well, many studies have now demonstrated that events at any point during a bird's full annual cycle are pivotal to driving subsequent events during the other parts of the year. So for instance, for our cute northern saw-whet owl, an event on the breeding grounds influences those events during migration and ultimately during the non-breeding season. So let's take a closer look at this through an example of the northern saw-whet owl. So as many of you may be familiar, saw-whets experience eruptive years where we see more hatch years migrating in a given year. And so people had observed this for a very long time, but nobody knew really exactly why, until researchers discovered that the influxes in northern saw-whet owls was directly related to the populations of red-backed voles on their breeding grounds. So this graph shows the voles with the dotted line and the saw-whets with a solid line. And you can see that these two populations are very, very closely linked. And so by noticing these things during migration, we're better able to understand what's going on in the rest of a bird's life cycle. So how do we create migratory connectivity? Well, we can do this in a variety of different ways. We can use extrinsic markers, so things like mark recapture, putting bands or tags on birds. We can also use electronic tracking devices, which have greatly improved over the years. We can also use intrinsic markers, so things that are inherent to the birds. So this includes things like DNA, looking at trace elements like lead or mercury, or using stable isotopes. So this is what I'm going to talk with you about today, is how we can use hydrogen stable isotopes to better understand raptor migration. So what is a stable isotope? Well, hydrogen comes in two different stable isotopes, protium and deuterium, and the only real difference between them is the presence of a neutron in the nucleus of deuterium. And essentially what this does is it makes deuterium twice the weight of protium. And because of this difference, these two isotopes act in very different ways in the environment. The main way that hydrogen is transported across a landscape is in the form of water. So as water moves across our landscape, these two different isotopes are deposited in different ratios due to those differences in weights. And so what we can see in this image, which is an ISOSCAPE, so ISOSCAPE is just short for an isotopic landscape. This is showing in different colors the different kind of, essentially you can think of this as a difference in ratio between these two hydrogen stable isotopes. And we get these really consistent patterns due to the biogeochemical processes that occur in our world. So what does this allow us to do in relation to migration? Well, hydrogen stable isotopes can be used to study and have been used to study migration in a lot of different organisms, including bats, butterflies, and birds. And this approach relies on the fact that you are what you eat, meaning that the isotopic signatures of the environment are taken up by organisms, and those same isotopic signatures or ratios are deposited into the tissues of those organisms. So we're interested in birds, and so we would be looking at the isotopic signatures that are present in the feathers of birds. So because we have these patterns across the landscape, what we can do to learn about the breeding origin of a bird is we can create a feather isoscape. So this requires taking known origin feathers and sampling them for deuterium, and then creating a kind of base map, a reference map, that can then be used to determine where an unknown feather sample was produced. So for my study, I have two objectives. First, to estimate the breeding origin of migrating raptors, and two, to identify those migration patterns that exist across the season. To do this, I'm looking at three different species: sharp-shinned hawk, northern saw-whet owl, and red-tailed hawks. So this fall, I was able to collect data from all three of these species. I collected 438 feather samples from sharp-shinned hawks, 423 from northern saw-whet owls, and 68 from red-tailed hawks. The next stages are to prepare the samples for hydrogen stable isotopes. So I'm going to, this fall and winter, clean, cut, and weigh these samples into small capsules to be analyzed. And then these feathers will be analyzed for hydrogen stable isotopes, and we'll use the results to answer our objectives. So for objective 1, assigning breeding origin, we'll take our unknown feathers and we will compare them to an established isoscape. We'll also constrain this assignment with current knowledge of breeding range and habitat, as well as any banding records that we have. For my second objective, which is looking at migration patterns, plan to regress our hydrogen stable isotope values against banding date, and also look at differences between sex and morphological characteristics. And so when we do this, we expect that we could see kind of three main things or combinations of those. The first is that we might find that southern populations are moving first. We could also find that northern populations are moving first through Duluth. Or finally, we might see that there's no real clear pattern, that we have some sort of broad front migration with raptors coming from many different locations all through Duluth at once. So I'd like to leave you today with some of the questions that we hope to answer with this research, and I hope that next year I can come and tell you what we found. So the first thing that we're interested in is, are early season migrants traveling from further south, or are they traveling from further north, or is there no such pattern? The second thing that we're interested in is, do males and females exhibit similar migration patterns? So for instance, we know for sharp-shinned hawks that females migrate before males, but are they migrating through Duluth with a similar pattern? In addition to that, do males and females migrating through Duluth come from similar locations? So are the early season migrants from females the same population as the first males that we see? Or are these two groups not coming from the same population? Are individuals from further north larger? So we're interested in learning something about the morphology. It's long been assumed that larger individuals are coming from further north, and we hope to see if this actually is true. And also, we're interested in, here at Hawk Ridge, the connection to the contaminants that we see in these organisms. So do we see a correlation between geographic origin and contaminant load? So those organisms with higher amounts of mercury or other substances in their tissues, are they coming from similar locations? And what can that tell us about those locations? And many more that I won't go into today. So just kind of to summarize, for this research, I've collected raptor feathers during the fall of 2020. They'll be analyzed for hydrogen stable isotope, and we hope to learn something about their migratory patterns and their breeding origin. So with that, I'd just like to say thank you first to my advisor, Dr. Matt Ederson. A big thank you to Frank Nicoletti, who is the banding director at Hawk Ridge, and a thank you to all of the Hawk Ridge staff and volunteers. I'd also like to thank the Integrated Biosciences program, and a big thank you to the grants that are funding this research, specifically to MOU's Savaloha grant, as well as the Hawk Migration Association of North America Research Award. So thank you so much for listening, and I look forward to your questions.